Okay, in this lesson we're going to look at reproduction. So this is booklet number 3 from the Foundation Year Pre-Med Pre-Dental, Semester 2. Okay, so reproduction begins with the fusion of paternal and maternal gametes, i.e. the sperm cells and the egg cells. The first thing that we're going to look at is spermatogenesis and then oogenesis. So this is the production of those spermatocytes or sperm cells and the ovum, the oocytes through oogenesis. If you just have a look at your notes though, on the first page of the notes, sexual reproduction involves the fusion of maternal and paternal gametes. These are haploid. When they fuse, firstly the sperm will release the nucleus into the secondary oocyte. When the two nuclei fuse, that forms the zygote. And we're going to concentrate now on how those haploid gametes are produced through these two processes of gametogenesis. So in males, this is spermatogenesis. In females, oogenesis. So let's begin then with spermatogenesis. It's important to note at this stage, the only structure that you need to know from this diagram is the seminiferous tubule. However, you may wish to label this diagram on the in your notes. So the tube from the testes, so this diagram takes a cross-section through the testes. From the testes there is a vas deferens, or two vas deferens, that will transport the mature spermatozoa from the testes to the urethra. Surrounding the testes is this epididymis. So the epididymis is where the spermatozoa are stored before ejaculation. Then we have the head of the epididymis, the vasa efferentia, which collect the spermatozoa from the seminiferous tubules before they're taken to the epididymis. The rete testes, the seminiferous tubules, which are the only structures that you need to be familiar with, and then the tail of the epididymis. This is a cross-section through a testes. Each of these circular shapes is, oh, that's not great, is it? Is a single seminiferous tube, or seminiferous tubule. It is possible to see in here the different stages of development of the spermatozoa. We are going to focus next on how the spermatozoa undergo meiosis to produce mature spermatozoa. One thing you might note, though, from these images, as we can see, these are the tails of the mature spermatozoa extending into the centre of each of these seminiferous tubules. That might give you a bit of an idea of the orientation of this diagram. Okay, you also have this diagram in your notes on page 1. It shows a cross-section through a testis. Firstly, labelled there are the Sertoli cells. Now the Sertoli cells are not involved in, do not become spermatozoa themselves, but instead are present to nourish the developing spermatozoa. Here we have the spermatids, the secondary spermatocytes, the primary spermatocytes, the spermatogonium, and then between the seminiferous tubules themselves are these Leydig cells with a basement membrane. So in the cross section through the testis, we can see the if I just draw on here, this is a mature spermatozoa. I'm just going to put this label in here. So mature spermatozoan. The development of those mature spermatozoan is from, firstly, it's beginning with the spermatogonium, then the primary oocytes, primary spermatocytes, secondary spermatocytes, the spermatids, and eventually the spermatozoan. The control of sperm production is by the luteinizing hormone, which is produced by the anterior pituitary. LH stimulates the production of testosterone by those Leydig cells that we showed in the previous diagram. In males, LH and testosterone are involved in a negative feedback loop. So, testosterone will control the rate of spermatogenesis. Sperm production usually takes place continuously and at a fairly consistent rate. Okay, you also have this diagram in your notes. This begins to show the maturation, or firstly meiosis, and then the maturation of the spermatozoan. This is the lumen of the seminiferous tubule, so that round space that we saw at the beginning, where I showed the tails of the sperm, or a spermatozoan. Here we have those Sertoli cells. Like I said, they provide nourishment to the development of the spermatozoan. And the spermatids. So spermatids are haploid cells, as are the spermatozoans. So we could label that, couldn't we? These are haploid spermatids, also haploid. The difference between a spermatid and the spermatozoan is differentiation, really. Genetically they are the product, not genetically, but they are the product of two meiotic divisions. Between the stage of the spermatids and the spermatozoan, the spermatids undergo differentiation, which is where they change in their shape from the circular or spherical cells into the typical sperm cell with a head containing an acrosome and the nucleus inside the head. We'll look at these in a bit more detail later. The middle piece of the sperm, which contains many mitochondrion, and the tail, which is comprised of microtubule proteins. It has a whip-like movement, making sperm motile. Below the spermatids in this diagram are the secondary spermatocytes. So the secondary spermatocytes are produced as a product of the first meiotic division. So these are also haploid. Before the secondary spermatocytes, we have the primary spermatocytes. Now, if we can see here that this is if this is the first meiotic division, or meiosis 1. During meiosis 1, I'll just note it down over here, the homologous chromosomes separate, and therefore, as they only contain one member from each homologous pair, the secondary spermatocytes will be considered haploid. The primary spermatocytes, well, they still contain both members of the homologous pair, and therefore they are diploid, 2N. Just to fill in that idea about meiosis, so between the secondary spermatocytes and the spermatids, this is meiosis 2. As we know, during meiosis 2, the sister chromatids separate. Not such an important label, this one, the tight junction between two Sertoli cells. But again, these are again an important one, so these are diploid, diploid spermatogonium. They are the product of mitosis from the germinal epithelium. So the germinal epithelium that surrounds the seminiferous tubule undergo mitosis to produce many spermatogonium. Those spermatogonium then individually undergo meiosis. Okay. In the let's get back to this. In your question booklet now, after you've labelled the diagram on page 2 of your notes, you may wish to have a go at question 1. Okay. Question 1 shows a flow chart of the main stages of the formation of the sperm cells in the testes. The diagram shows a dividing cell from a human testis. Only 4 of the 46 chromosomes are shown. So at this stage, I would suggest that you pause the video, and after you've paused the video, have a go at doing this question. Give yourselves 5 minutes or so to do that, and then you can return to the video, and I'll go through the answers to this afterwards. So pause now. No cheating, and you can have a go at doing this question. Okay then, let's have a look at the mark scheme for question number 1. In fact, back to there. So the question asks, during which of the stages A to E in the flowchart would this division occur? So we need to look at the evidence from this diagram, and in the diagram we can see that each chromosome exists as a pair of sister chromatids joined by a centromere. Those sister chromatids are the product of DNA replication. Now we can see that the homologous pairs are being separated at this stage, and therefore, if these homologous pairs are being separated, that must be meiosis 1. We can be a bit more specific, and we know that the homologous pairs are being pulled apart as the spindle fibers shorten, pulling those homologous chromosomes apart. So we can say that this is anaphase of meiosis 1, or anaphase 1. As we said from the diagram before, meiosis 1 occurs between the primary spermatocytes and the secondary spermatocytes. So this must therefore be stage C. The reason for that is that the homologous pairs of chromosomes are being separated towards the poles. So if I look at the mark scheme for this, stage C, we can see the formation of a secondary spermatocyte. Evidence for that, it shows, well, we can have any of these, shows anaphase of meiosis 1 or anaphase 1. Perhaps the most important evidence here is that it shows the chromosome number being halved, or perhaps I would prefer the homologous chromosomes being separated for question number 1 there. Okay, so let's go back to the PowerPoint and we'll continue. Okay. Sorry, I'm navigating question number 2.1 as well. The diagram shows the main stages of the formation of sperm cells in human testis. Describe 2 ways, other than size, in which the cells at anaphase of division A would differ from those cells at anaphase of division B. If you'd like to pause your video now, you can have a go at doing question number 2. Okie doke. So, question number 2A. The homologous chromosome pairs would be separated during anaphase, whereas anaphase 1, whereas chromatids are separated during anaphase 2. Those diploid cells in anaphase 1, whereas they will be haploid cells in anaphase 2. During anaphase 2, the centromeres would be separated, but not during anaphase 1. Part 2b, let's look at the diagram there. Explain 2 ways in which meiosis contributes to genetic variation of the mature sperm. So this is a recall really for what we did with meiosis. 2 ways. So crossing over will mix and give new combinations of alleles. Independent segregation mixes and produces new combinations of maternal and paternal chromosomes. Okay, so that is spermatogenesis. We'll next look at oogenesis in the ovaries. So, oogenesis then is the differentiation of the ovum by processes of mitosis followed by meiosis. This is a secondary oocyte. So the secondary oocyte is the structure. It's more than just one cell that is released during ovulation. Okay, so what you have in this diagram, from the outside to the inside, we have the remains of the follicle cells. So these were those follicle cells, the granulosa cells that were present in the Graafian follicle that we'll talk about later. Inside of that is what's called the zona pellucida, which is a protective jelly-like layer. Inside that is the cell surface membrane of the secondary oocyte. The secondary oocyte contains cytoplasm, a large cytoplasm with many food reserves, many lipids and proteins necessary for the development of the zygote after fertilization. You may have noticed between the zona pellucida and the cell surface membrane is what's called a haploid polar body. Now this is the product of meiosis 1. So during the two meiotic divisions in oogenesis, the cytoplasm is unevenly distributed, with all of the cytoplasm being directed towards one cell. The nuclei are still divided, but that produces one large cell after meiosis 1 and one small cell. That small cell is known as the polar body. It may be degraded or it may remain underneath the zona pellucida, but it will not develop into a secondary oocyte. During meiosis 2, the same thing happens again, so a second polar body will be produced. Okay. So follicular genesis is the development of the follicle. The primary role of the follicle is to support the oocyte. At the beginning of follicular genesis, around 40 follicles may begin to develop, and they compete for dominance. They compete for the oocyte that will be released during ovulation. Around 20 will reach maturation, but only one ever is released or reaches ovulation. Lasts about a year, roughly, just over a year. And at any time, the ovary may contain follicles at all stages of development. Here we can see the ovary. This is a fairly typical diagram that you might expect to see of the ovary, and you have a similar one in your notes. So the ovary, or this diagram, demonstrates that cycle, the follicular genesis, all the way up to ovulation, and then the development of the corpus luteum and the degeneration of the corpus luteum after ovulation. Ovulation releases the secondary oocyte from what's called the Graafian follicle. So the Graafian follicle is the mature follicle containing the secondary oocyte. Okay, you have this diagram in your notes that you may wish to label. So surrounding the ovary is the germinal epithelium. These are diploid somatic cells that undergo mitosis to produce primordial follicles and primary oocytes. The primary oocyte, shown in the centre there, is the darker area. Let's see if I can show that in here. So that's the darker area within the follicle, with the primary follicle continues to develop to form a secondary follicle. The secondary follicle again continues to develop to form the Graafian follicle. Now this is the mature follicle from which the secondary oocyte will be released at ovulation. After ovulation, the secondary oocyte is released, surrounded by those granulosa cells. Within that is the zona pellucida. Inside that is the secondary oocyte itself. After ovulation, the Graafian follicle that has been ruptured will develop into an active corpus luteum. Now, depending on whether or not the secondary oocyte completes meiosis and is fertilized, or if that secondary oocyte is not fertilized, if it is, the active corpus luteum will remain. And it secretes progesterone during the earlier stages of embryo and fetal development. If it's not, if the secondary oocyte does not complete meiosis and is not fertilized, the corpus luteum will be degenerated and will break down. The structure, or the sort of the area within the ovary, is known as the stroma. This diagram demonstrates the stages of development in the formation of the gamete, or the mature ovum, shown down here. Okay, so similar to the spermatogenesis, germ cells undergo mitosis to produce, this time, oogonium. Oogonium continue to divide by mitosis, so we get this multiplication taking place in the ovaries of the female fetus. So this stage, this multiplication phase, now you don't need to refer to it as the multiplication phase, but you may wish to know this. This takes place in the female fetus, so before birth. So the production of these oogonium takes place before birth. Once the female reaches puberty, or after puberty, one of those oogonium will undergo meiosis. In fact, many of them may begin this process, but only one will actually reach the final stage. So each month after puberty, the oogonium undergo this growth phase to produce the primary oocyte. Now the primary oocyte is still diploid. That diploid primary oocyte undergoes meiosis 1. If we just remind ourselves, meiosis 1, we get the separation of homologous pairs during meiosis 1. So in the two cells that are produced, we have a small first polar body and a much larger secondary oocyte, which is now haploid. So as I said before, the primary oocyte, when it divides, the cytoplasm is unevenly distributed, with the vast majority of that cytoplasm going into the secondary oocyte. At this stage, the secondary oocyte pauses meiosis. The secondary oocyte continues at this just after meiosis 1 is completed, The secondary oocyte is the oocyte that is released during ovulation. Meiosis 2 will only be completed after a sperm fuses with the membrane of the secondary oocyte. The sperm will release its nucleus, its haploid nucleus, into the secondary oocyte. This is referred to then as the male pronucleus. The female pronucleus within the secondary oocyte is then stimulated to complete meiosis 2 to produce the haploid female pronucleus. At this stage, there will be a production of a second polar body. The male pronucleus then fuses with the female pronucleus. We get so the nuclei fuse to produce the diploid zygote. So just have a look at the numbers here. After conception, the ovary will contain 4 to 5 million follicles. Sorry, this is the conception of the individual which will later undergo oogenesis, not the zygote that I've just mentioned previously. By the time a female reaches puberty, there will be between 25,000 and perhaps 1.5 million follicles. Typically, about 400 of those will reach the pre-ovulatory stage, with approximately 1 release per month after puberty. This diagram again demonstrates what we looked at previously. We've got those oogonium, the diploid oogonium, which undergo mitosis many times to produce many of these diploid oogonium. Now this is prenatal, so this is all taking place within the female fetus. During the childhood years, in infancy, the oogonium persist at this stage, so prior to or part of the way through meiosis 1. When they reach puberty, meiosis 1 is completed. That produces the secondary oocytes and a haploid polar body. Sorry, no, that's not a secondary oocyte. That is a primary. Yeah, okay. So these are oogonium. Oogonium undergo meiosis 1 to produce the am I right here? Yeah, to produce the primary oocyte. Meiosis 2 begins, but then is arrested, paused, at metaphase 2. Here we have the secondary oocyte. That secondary oocyte is what's released during ovulation. Meiosis 2 is only completed when the nucleus from the sperm, that haploid nucleus of the sperm, enters the cytoplasm of the secondary oocyte. At this stage, meiosis 2 is completed. Those two nuclei fuse to produce a diploid zygote. Okay. So here we have the the primary oocytes in the primordial follicle. So this is number 2 on your diagram in your notes. Sorry, just realised what I've done. Let me go back a stage. Let me erase these. Oh dear, what a silly error. Okay, so these are the secondary oocytes. Up here we have the primary oocytes. Okay, so the primary, of course they are. Those primary oocytes are diploid. They undergo meiosis 1 to become haploid. Oh, how stupid. To form the secondary oocytes. It's the secondary oocytes which are present in the Graafian follicle. When that Graafian follicle ruptures to release the secondary oocytes, they may be fertilized, and that fertilization is when meiosis 2 is completed. Okay, next we will look at the Oestrous cycle, so the control of the ovulation. So in humans, the cycle lasts around 28 days. Usually a single ovum is produced from one of the 2 ovaries. The cycle is responsible for coordinating the steps in the ovum production and preparing the endometrium. So the endometrium, the lining of the uterus, and that's necessary in case that, that secondary oocyte fuses with the nucleus from the sperm to produce a zygote. Okay, so we need to be familiar with these 4 hormones. FSH, or follicle stimulating hormone, LH, luteinizing hormone, both of which are produced in the anterior pituitary gland. In the ovaries, we have the 2 hormones, estrogen and progesterone, produced from the ovaries. You have this diagram in your notes, and I'm going to take you through the different stages of the Oestrous cycle and the roles that each of the hormones has in controlling that. Okay, so we'll begin with FSH. FSH is responsible for the development of the Graafian follicle. The Graafian follicle shown here. Oh, where's my pen gone? Let me go back. Okay, so this is the Graafian follicle, and it's that Graafian follicle that contains a secondary oocyte. The Graafian follicle contains granulosa cells, that are responsible for producing estrogen. So as the Graafian follicle develops, more and more estrogen will be produced, which is why we see this increase in the blood levels of estrogen as the Graafian follicle continues to develop. Estrogen has the functions of repairing the uterus lining, the endometrium, after fertilization. It also stimulates the production of FSH and LH by the pituitary. But it only does that when there is a high concentration of estrogen. So what we can see here is initially a low concentration of estrogen. We can imagine that this is sort of the threshold. A low concentration of estrogen will inhibit the FSH and the LH. Once it reaches this this critical concentration, there is a positive feedback and the concentrations of FSH and LH will increase. Down here we can see the uterus lining, the endometrium, increases in thickness as the concentration of the estrogen also increases. LH, luteinizing hormone, is what causes the Graafian follicle to rupture. The Graafian follicle then develops into the corpus luteum. So LH also stimulates the corpus luteum to produce progesterone. Okay, if we look at the diagram there, then we can see that the LH reaches its highest concentration just prior to ovulation at around 14 days. So it's the LH which is responsible for rupturing that Graafian follicle. We can see after the Graafian follicle is ruptured, it develops into the corpus luteum. That active corpus luteum produces progesterone. So we can see the concentration of the progesterone is greatest after the Graafian follicle is ruptured. Progesterone is responsible for maintaining the thickness of the endometrium, the lining of the uterus. It also inhibits the production of LH and FSH from the pituitary gland. So we can see when the concentration of the progesterone is high, the concentration of the LH and the FSH is at its lowest point. This is to prevent further follicles from developing and further ovulation, because the progesterone's role in maintaining the thickness of the endometrium is to allow implantation to be successful. It wouldn't be sensible if a second Graafian follicle ruptured to release a secondary oocyte during that time. Okay, so we have some questions here. The 4 hormones that regulate the Oestrous cycle: FSH, LH, estrogen, and progesterone. Which ones are gonadotrophins? So these are the hormones produced by the ovaries. So they are estrogen, I can't spell, there's an R in there, and progesterone. Which hormone stimulates the follicles to develop? Follicle stimulating hormone. Which causes the lining of the uterus to build up, become thicker? Estrogen. Which hormone is released from the Graafian follicle? Estrogen again. Which causes the Graafian follicle to rupture? Is luteinizing hormone. Causes ovulation, again, luteinizing hormone. Maintains the thickness of the uterus, is progesterone. Released from the corpus luteum, progesterone again. Which hormone stimulates the estrogen to be, I don't know what that says at the moment. Estrogen to be produced, perhaps. Might be FSH. Okay, so there are some negative and some positive feedbacks within the Oestrous cycle. LH stimulates the rupturing of the Graafian follicle and the development of the corpus luteum from the Graafian follicle. The corpus luteum then produces progesterone. There is a negative feedback then by the progesterone on LH. FSH, which is responsible for the development of the follicles and the Graafian follicle, stimulates the production of estrogen. At low concentrations of estrogen, there is a negative feedback on FSH. So perhaps during those first 10 to 12 days of the Oestrous cycle, when the concentration of the estrogen is below a critical concentration, there is a negative feedback. However, after perhaps day 10, 11 and 12, when the estrogen concentration is higher, it reaches a critical threshold. High concentrations of estrogen have a positive feedback effect to release FSH just prior to ovulation. So we'll take you through all of that once more. FSH is released from the anterior pituitary to stimulate the development of a follicle and the maturation of a Graafian follicle. As those follicles develop, estrogen is released. That estrogen maintains or builds up the thickness of the endometrium after menstruation and has a negative feedback on the release of any further FSH at low concentrations. We've got that negative feedback at low concentrations. LH is released when that estrogen concentration becomes higher. LH causes the rupturing of the Graafian follicle. The Graafian follicle develops into a corpus luteum, again stimulated by that LH. The corpus luteum then produces progesterone. Progesterone maintains the thickness of the endometrium, but has a negative feedback effect on the release of any further LH. When the corpus luteum breaks down, progesterone is no longer secreted. So that would be corpus luteum would break down if the secondary oocyte is not fertilized. At which stage there's no longer any inhibition of the LH. There's no longer inhibition of the FSH. Menstruation follows, and the process and the cycle is repeated. Okay. So I'm not going to talk you through these stages, but if you want to stop the video to label your diagram here, you can. Okay, so that was the control by those 4 hormones of the oestrous cycle. You may need to go back through the video and pause it at each stage if you'd like to annotate your diagrams. This slide is just a little interesting one. With addition to all of those other levels of hormones and the thickness of the endometrium, etc., there's an extra a row here showing the basal body temperature. Now, just prior to day 14, we can see that there is a big increase in the basal body temperature. Question here, why might knowing this be useful? Well, it has been used as a method of contraception, but I wouldn't say it's the most reliable method of contraception. There are apps that you can get on your phone to monitor that. Perhaps it's more beneficial to know if you want to get pregnant that there will be an increase in temperature, basal temperature after ovulation. Okay, here we have the pituitary gland showing the anterior on the right, posterior on the left. Okay. After spermatogenesis and oogenesis, fertilization may follow. Here we have the structure of the sperm. So, in the head, there is an acrosome. These contain hydrolytic enzymes. The nucleus, the centriole, which is responsible for producing the microtubules within the tail of the sperm. There are those central microtubules. In the middle piece, this part here, behind the head or between the head and the tail, there are many mitochondria necessary for the whip-like movements of those microtubules in the tail from sperm motility. Here is a diagram of the secondary oocyte. We have the nucleus within. Remember that nucleus has not yet completed meiosis 2 and will only complete meiosis 2 when a sperm enters. We have these circular structures, lysosomes, referred to as the cortical granules, close to the membrane of the oocyte. Then the cytoplasm, containing many lipid droplets. There are microvilli all around the surface of the membrane. They are able to absorb nutrients from the follicle cells. Those follicle cells, or the granulosa cells that surround the egg, are also responsible for producing this jelly-like coat called the zona pellucida. Within that zona pellucida, there may be the first polar body. So remember that smaller cell produced after the first meiotic division. Here we can see the relative sizes of a secondary oocyte and a spermatozoan. Okay, the key points of fertilization. First, there is specificity. So receptors on the membrane of the secondary oocyte, which are specific to proteins on the surface of the sperm. Hydrolytic enzymes are released from the acrosome, which are able to digest through the zona pellucida. The haploid sperm nucleus meets the haploid oocyte nucleus. Oocyte completes meiosis 2. The two nuclei, or the two pronuclei, fuse to form a zygote. Okay, this next slide, or the next couple of slides, we're going to look at the cortical reaction. This is necessary to prevent more than one sperm entering the oocyte. During the cortical reaction, enzymes are released from the cortical granules that have two roles. Firstly, they hydrolyze the sperm receptor proteins, preventing any more sperm from binding. And they cause the zona pellucida to become thicker and impermeable to sperm. This diagram demonstrates the whole process. So first we have, on the left, number 1, contact. When the sperm head comes into contact with the zona pellucida, that stimulates the secretion of hydrolytic enzymes from the acrosome onto the zona pellucida, and we get this acrosomal reaction where the hydrolytic enzymes from the acrosome digest a pathway through the zona pellucida. At this stage, there is the growth of the acrosomal process. So these are proteins present in the head of the sperm, which extend and bind to the receptor proteins on the surface of the secondary oocyte. This is that specificity, ensuring that only sperm from the correct species will be able to gain entry to the secondary oocyte. After that acrosomal process takes place, the acrosomal reaction and the binding of those proteins, the nucleus of the sperm is released into the cytoplasm of the secondary oocyte. That stimulates the cortical reactions. Those lysosomes containing those cortical enzymes fuse with the membrane of the secondary oocyte. Those cortical granules thicken the zona pellucida and hydrolyze the proteins, the receptor proteins on the membrane. That prevents any further sperm from binding. Question 3 in your question booklets now, you might want to have a look at that one. So if we have a look at question 3, I want to pause the video at this stage to look at question 3. Here. So first we have this graph showing the concentration of hormones responsible for the control of the menstrual cycle. Okay, so have a go at doing question number 3 for me, and then we'll look at, lastly, we'll look at the development of the fetus. Okay, question number 3 then. This is the mark scheme. Pause the video at this stage. Those hormones that you needed to label first were C and B. The role of FSH is to stimulate the release of estrogen. Initially, estrogen will inhibit FSH secretion. Just remember, estrogen inhibits at low concentrations. We get that negative feedback at a low concentration of estrogen, positive feedback later. Okay. So in 3.4, the increase in estrogen after days 10, 11 and 12 to that critical peak, or to the threshold concentration. And here we have the positive feedback at high concentrations of estrogen, stimulating the release of more FSH just prior to ovulation. Okay, after fertilization, there is implantation of the, firstly the morula that's produced, the ball of undifferentiated cells. That begins to differentiate into a blastocyst, and the blastocyst binds or attaches to the endometrium during implantation. Later, as differentiation continues, we get the development of the embryo and the placenta. Okay, so the layer surrounding the embryo is known as the chorion. The chorion, with their produce a chorionic villi. Those villi increase the surface area and break down the endometrium to allow for more successful exchange between the chorionic villi and the endometrium. Here we can see the umbilical stalk. The placenta will develop from the chorionic villi. The embryo is within the amniotic cavity. The amnion is a sac that surrounds that amniotic cavity, and then there is a yolk sac within the embryo there. Okay. So, surrounding the blastocyst is a layer of cells, a surface layer of cells called the trophoblast. It is those trophoblasts which produce the chorionic villi and will eventually develop into the placenta. They secrete human chorionic gonadotrophin, a hormone. Is HCG, which is the hormone detected in a pregnancy test. Without fertilization, the corpus luteum will degenerate. Progesterone concentrations decrease, the endometrium breaks down, causing menstruation. LH is responsible for maintaining the corpus luteum. When or after implantation, as the human chorionic gonadotrophin HCG concentration increases, it has a similar function to LH, so it will maintain the corpus luteum during the early period of implantation. Later, the placenta will produce progesterone, and that progesterone will maintain the thickness of the endometrium. As I said earlier on, the chorionic villi and the trophoblast release enzymes. They break down blood vessels within the endometrium, and we can see the maternal blood moves into these pools surrounding the chorionic villi. That increases the contact between maternal blood, which is within those pools, and the capillaries of the chorionic villi, which will eventually become the placenta. These pools, those spaces, are called lacunae. that surround the chorionic villi. It's important to remember that maternal and fetal blood do not mix, so the placenta is adapted for exchange. It's got a large surface area, short distance of diffusion. There is a gradient for exchange, or there may be active transport. And here is a diagram. Here is an image of a placenta shortly after birth. These next few images just show the stages of fetal development. So at 5 weeks, 8 weeks, etc. The fetal circulation is adapted. This means that the maternal organs play the function of some of the fetal organs during the period of gestation. There are 3 structures that you need to be familiar with. The ductus arteriosus, the ductus venosus, and there's another one somewhere. The foramen ovale. Okay, so these are ways to redirect the blood during fetal development. You need to be familiar with these 3 adaptations. So, very little blood travels to the lung tissue. The foramen ovale, the hole in the heart, diverts blood from the right-hand side of the heart into the left. If we go back to the diagram here, we can see the foramen ovale, which diverts, where is it? Okay, blood from the right-hand side of the heart into the left. The pulmonary artery is diverted into the aorta by the ductus arteriosus. Go back to that diagram again. Here we can see the pulmonary artery here. Blood is diverted directly into the aorta, so that prevents blood travelling to the lungs. Then lastly, the role of the liver is performed by the mother's liver. So the umbilical vein is connected to the inferior vena cava and therefore bypasses the liver. This is called the ductus venosus. Here we can see the ductus venosus, so the umbilical vein directed to the vena cava. So it's just these 3 adaptations that you need to be familiar with. Okay, and that is everything on reproduction and development of the fetus.